So far, we have only focused on the first part of the secret of life. How, when the chromosomes divide, information is passed from one generation to the next. But the other big question is how that information is turned into flesh and blood. This is where the concept of a gene enters the story. A gene is simply a length of DNA code letters that carries the instructions to make a protein. Proteins are basic components of our bodies. They are the main ingredient of obvious structures, such as our eyes, our skin, our hair, and our muscles. Proteins also play a hidden but highly important role. As enzymes, they act as molecular machines that carry out numerous chemical operations in the cell. That helicase copying DNA at the speed of a jet engine, it's an enzyme. There are thousands of different enzymes that scurry around our cells, performing all kinds of tasks. Enzymes are the miniature engines of life. They are made of protein. So the second part of the secret of life is that the DNA code is no more and no less than a set of instructions to build proteins. Consider the blood system that keeps our body supplied with oxygen. The heart pumps blood through the lungs where it picks up oxygen. The oxygen is carried in the red blood cells by a highly specialized protein called hemoglobin. In this red blood cell, there are 280 million hemoglobin molecules, each ready to do its job as an oxygen transporter. So how does the DNA make a protein such as hemoglobin? Proteins are made of building blocks called amino acids. There are 20 different kinds. Linked together in chains, they form proteins. It's the precise order of amino acids that determines what kind of protein is made and what it does. So the next big challenge was to work out how the 4 letters of DNA could code for each of the 20 amino acids that make protein. Back in the 50s, it seemed a tough nut to crack. The first question was, how many DNA letters coded for each amino acid? If it was 1 DNA letter for 1 amino acid, then you could only code for a maximum of 4 amino acids. 2 letters in every possible combination could code for up to 16 amino acids. Still not enough. But 3 DNA letters provide more than enough combinations to code for all 20 amino acids. So 3 was the answer. It was a triplet code. However, scientists still didn't know how these triplets of DNA letters could produce a string of amino acids.
I thought the problem would last me my lifetime. I had no idea it would be years.
What you are about to see is DNA's most extraordinary secret. The innermost workings of how a simple code is turned into flesh and blood. This is what Francis Crick called the central dogma of modern biology: how DNA makes protein. It starts with a bundle of factors assembling at the start of a gene. It's these that trigger the first phase of the process, reading off the information that will be needed to make the protein. The gene is the length of DNA stretching away to the left. Everything's ready to roll. 3, 2, 1. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the 2 strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to make an exact copy of the A's, C's, G's, and T's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related nucleic acid known as U. You are watching this process, called transcription, in real time. It's happening right now in almost every cell in your body.
It's a bit like taking the information off a hard disk on a computer and put it into memory, making it a real program that's running. So that process of DNA to RNA is like running a program, double-clicking on an icon.
When the RNA copy is complete, it snakes away from the nucleus and into the outer part of the cell. Then, in a dazzling display of choreography, all the components of another molecular machine lock together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids. They all carry a specific 3-letter code that will be read by the machine. Now we come to the heart of the process. Inside the ribosome, the RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecule. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic message you feed in on the RNA. In this case, the end product is hemoglobin. The cells in our bone marrow churn out 100 trillion molecules of it per second. And as a result, our muscles, brain, and all the vital organs in our body receive the oxygen they need. Unless something stops the